Hello everyone, welcome to Biology 330, Mammalian Physiology. This is the virtual lecture for Chapter 2, what we're calling Chemical Physiology. This is giving you the chemical understanding that's important for understanding the larger role, particularly of proteins. Proteins are where it's at when it comes to almost everything that we do in the human body. It may not seem like that when you're splinting an arm or you're doing some type of physical therapy with someone, but the truth is what you're doing is modulating protein function. So we need to understand this in order to be able to have better access to the mechanisms and so forth that we're going to talk about down the line. So with this lecture, as well as all of my lectures, please take good notes. It's not enough just to follow along. If you just follow along, you'll probably find yourself having to go back and repeat the exercise again. There's about 50 slides in this presentation, so I anticipate it taking a little longer than an hour. So take good notes, look for points of emphasis, see what I'm circling, see what I'm repeating, and that will help give you sort of a priority list when it comes to preparing for the exam. So I have some learning goals here for you, so I'm going to just scroll these in. This is sort of like a study guide for you. So consider these as task lists, not something to be minimized, but as a starting point for greater understanding. So levels of protein structure, understand that role between shape and function. Sometimes we say structure and function. Structure and function are everything in biology and particularly physiology. So we're going to talk about pH today. We're going to talk about the reasons why pH and other environmental conditions are extremely important for physiology, for therapy, for medicine, and for life. And we'll be able to recognize the different functions of proteins. We're also going to show protein interactions today. Understanding protein interactions is the key to understanding things like pharmacology and the key to understanding how healing as well as injury works. Okay? How are these protein interactions influenced? And so we have a bunch of terms here. So this is relatively straightforward. Cofactors, activators, modulation, competitive, irreversible. These are all just terms that will help you become more conversant. Types of inhibition. This is really important when we're talking about blocking protein function. Like for instance, when we put ice on an injury, what we're doing is inhibiting protein function. We're inhibiting inflammation. Talk a little bit about reaction rates. Reaction rates are how we study protein function, so it's good to understand reaction rate from a clinical perspective to understand how we know when we have a new drug or how we know when a drug is working and so forth. Before we start, I've sort of already alluded to this. We're talking about chemistry today. It's the chemistry behind physiology. It sets you up. Yes, there's a bunch of review today, and if you've just had chemistry last semester or over the summer, some of this will feel like old hat. I'm going to be putting some mastering A and P up to help refresh you on this. If it's been a number of semesters since you've had chemistry, it's good to pick that stuff back up, at least for now. Maybe you're not going to be involved in pharmacology as much, but that doesn't mean that you shouldn't have an awareness of it. So we're going to be dealing with this over the next few chapters, so it's a good review situation. Comfortable with ions. Everything that sort of happens in the aqueous environment of our cells is ionic in nature. And so we're regulating this movement. We're regulating the transport of these ions in and out, across, through, and so forth. Logarithms and pH, I'll just allude to that a little bit. It doesn't have to be as hard as it might seem. Talk about acid bases and buffers. That's really, really important for the structure of proteins. And we're going to talk a little bit about the biomolecules. I'm going to be hitting this up more in the lab next week. We're going to be using some examples, showing you some things. And you've seen some of this before, I'm sure, in 155 perhaps, definitely in 165. What makes this and all this material that we're going through just so important? Why spend the time on this? Well, first of all, 99.9999, I mean literally every single thing that happens in your body, every single thing that happens in your body probably involves some protein at some level, and more than likely uses a whole bunch of proteins, thousands and thousands of them, maybe thousands of the same kinds of proteins, maybe whole families of proteins. Proteins are the doers. They are the ones that really get the job done. Maybe DNA helps store information, maybe RNA might help transmit information, it might be somewhat catalytic in nature, but this is where the action is, and so understanding protein function is the key. It is the gateway to therapy. It is the gateway to medicine. Okay? Proteins do all sorts of things. They may be relatively benign, not to say benign, but non-functional. What I mean by non-functional is they're not catalyzing something. However, they may serve a structural role. So the creation, the maintenance, the degradation in some cases is very important to understand. Proteins are signalers, like insulins and hormones. They signal cells to grow or not to grow. They catalyze metabolic reactions, like the creation of molecules and the getting rid of molecules from the body. Antibodies for immunity and cell proteins for antibody activation and creation. Making cells what they are. For instance, a muscle cell is not the same as a skin cell. So we have to regulate which proteins are expressed in order to give cells their differing functional roles and differing identities. Proteins serve as toxins, okay, in spiders and venoms and so forth, and in transport. There's all sorts of things out there. So what makes this important is if you are going to be involved in helping people with injuries. Most injuries involve some damage to a protein, like a structural protein in the case of a muscle tear. And protein function is required to minimize further damage, for instance, or when you're healing. So understanding how proteins do what they do in order to facilitate healing. Why is it that when somebody has an injury, it makes sense to put a little bit of ice on initially, but you don't just bathe them in ice for hours and hours and days? Why is that? The reason is that at some point you're going to need protein function to repair and restore. So ice is inhibiting protein function, so you might actually slow healing if you ice it too much. You may even cause injury if you ice it too much. So interventions, activities, adjustments to the body in order to prevent or restore function in the case of loss of function. We're going to block or slow down protein functions like ice. Okay? Sometimes we're going to stimulate protein functions like with heat or massage or what we call a tense unit, that's electrical stimulation. Or we might use medicines. We might have therapies that are prescribed, or pharmaceuticals, drugs, are selected to modulate protein functions in the body. So we're going to modulate functions by modulating proteins. That's why this is so huge, and that's why I spend the first section talking about cellular biology, because this is really where it's at. There's a link here. I'm going to allude to this later. I'll just hang tight on that. I'll put it here now, but you'll see this link again later, and I'll explain what that is. And it's just an example. This is from a cancer paper showing how the disease genome in cancer creates screwed up proteins, and that leads to disease in the cells. We understand protein sequence. We may be able to find new drugs to modulate those proteins, like right here. We might be able to identify new ways of detecting cancers or triggering the immune system based on the proteins that tumor cell is expressing. And we may be looking for responses. We may be able to better understand what the body is doing so that we can adjust the body in order to do different things in order to make the therapies more effective. All comes back to proteins. All comes back to proteins. So this chapter is the foundation. I trained as a pharmacologist, so I think this is really where it's at, that we're laying the foundation to give you the tools to do great things down the line. You have awareness here. It's going to make everything that you do down the line so much easier. So let's do a little bit of a case study here. So I'm showing you this now, so we're going to get into some more of why the chemistry is important. So why, if we look at this panel up here, this is called a Chem 7, or if we include calcium down here at the bottom, they call it a Chem 8 panel. So we have things like sodium ions and potassium and chloride and bicarbonate, the amount of nitrogen, creatinine, glucose, and calcium. Why are these so important? Well, if you've listened for the few slides before, it's because they influence protein function. Okay, when you see these in the clinic, you'll often see them broken up in these little fish skeleton type charts. Okay, and if we could, look at a full report, we might have our Chem 7 right here, along with blood gases. And guess what? pH. Okay, PCO2. Okay, why are those important? Gets back to protein function. You don't have protein function, you don't have life. So ions, pH, gases, all influence protein structure and function. Okay? pH is a huge one. pH is really important. We're going to show you why and how pH influences protein structure today. Okay? So ions, pH are huge in protein. Okay? If we look at all of the structures in a cell, they are comprised of a combination of usually lipids, maybe a little bit of carbohydrates on the outer membrane, but proteins, and proteins. So this is a picture by a guy named David Goodsell, who's a scientist and an artist. And what you see here is a membrane here, and in that membrane there are all kinds of proteins. The letter A here, that's a protein. Letter B here, that's a protein. Letter C here, that's a different protein. So proteins are very, very, very important in both the structure and subsequently the function of the cells. And the cells are the basic unit of life. So when we deal with life, we're dealing with cells. Okay? And cells depend on proteins. Have I made my point? I've probably done it in about 6 or 7 slides here. I don't want to beat the dead horse, but I'm trying to help give you some visual appreciation for why we invest this time. Okay, so proteins are sitting right here in this figure, right here. Yes, so there are other macromolecules. We have carbohydrates, we have lipids, we have nucleic acids down here in what we call the genome, so the DNA down here. The transcriptome is that we're not taking every single DNA sequence and turning it into proteins all the time. What we do is we take a subset depending on the cell type, the cell function, if it's under stress, if that sort of a thing. So that's called the transcriptome. So RNA would be right here. We turn most of those RNAs into proteins, and then the proteins are what do most of the work, maybe with the help of carbohydrates and lipids over here for structure. Okay, but we're acting at this level today. Just another figure. I'll breeze on past this, but just to show you again where proteins are involved, why we think they're so important. So about this chapter today, we're going to deal with the chemistry side of it, molecules and bonds and the properties of atoms, and the non-covalent interactions that I'll discuss a little bit more later on. These then lay the foundation for the biochemistry, which leads to protein interactions. Interactions is another way of saying functions in some cases. Many times you need protein interactions in order to achieve function. So molecules and bonds, this should be stuff that you already understand, that bonds are ways that atoms are linked together. They can involve electron transfer, or sometimes they involve electrostatic charge. They can be used for structural purposes, like this fat molecule here. This is a triglyceride. It's a lipid. Okay. They can involve structure, but they can also be used to store energy and transfer energy in some cases. Why is it that fats have so many calories? The reason they have so many calories is that each one of these lines here is a chemical bond from which energy can be harvested. Okay? So when you're burning fats, let's say you're out on a long run and you've been running for more than 20 minutes and you're clearly in an aerobic situation. That process of turning fats into glucose to create ATP is known as beta oxidation. So if this is the alpha here, this carbon atom right here, we're going to oxidize the beta, and we're going to bust this molecule, this little piece off. Or maybe this little piece off. So it's called beta oxidation. Requires a lot of oxygen to make ATP. That's where aerobic exercise, that's what aerobic exercise is doing. That's why aerobics are so good for burning fat. So we're going to talk about true bonds that involve electrostatic and electron exchange versus what we call weaker interactions, non-covalent interactions. And they're important for things like lipids versus aqueous things and so forth. So we'll talk a little bit about that. Okay? But we're storing and transferring energy with bonds. That's a big, big thing. It's not just about structure. It's also about what those bonds can do. So let's take it back a little further. So bonds involve electrons. Okay? Bonds involve electron function. And so there are 4 major functional roles for electrons in physiology. 4 major roles. The first one is the formation of covalent bonds, again for energy storage and for structures. The next important one is ions. Okay, the formation of ions. Ions where you have either an extra electron or you're down an electron versus the protein have very, very important roles in physiology. These things called high energy electrons, and I'll get to that in a little bit. That electrons can have different energy states, and just like the fluorescent bulbs that glow over your head in a classroom, or the sun, the light emanating from the sun, all derives from electrons being kicked up in energy state and then giving off that energy as light. And then lastly, free radicals down here. We hear about these sometimes when we talk about antioxidants and so forth. I'll get to those in a little bit. Okay? So covalent bonds, the formation of structures, the formation of reactive groups, and the storage of energy. Ions. They serve aqueous roles for things like gradients and for reactions in the body. High energy electrons, again, for storage of energy within an atom, not just across atoms like up here, but actually within an atom. And then lastly, free radicals, and these sort of coexist with these high energy electrons. It has to do with the reactivity of the atom and the amount of energy that's present to form bonds and so forth. We'll go to each of these in turn. So let's start with covalent bonds. Covalent bonds are where atoms share a pair of electrons, and they don't share it evenly all the time. If it was 2 carbons here, the carbons would have the same affinity for those electrons, and so those electrons would be evenly distributed across both atoms. However, carbon has what's called greater electronegativity than, in this case, hydrogen. So the cloud of electrons over the carbon is actually a little bit bigger. You know why? Because that electron that may have come from the hydrogen is spending a little more time with the greater affinity carbon. Okay? So they are sharing a pair of electrons in a covalent bond. It just may not necessarily be equal, and that's what polar and non-polar. We'll talk about that in a second. It can share 1 pair of electrons in a single bond, 2 pairs of electrons, so a total of 4 electrons in a double, or even 6 electrons, 3 pairs in triple bonds. And like I said, if there is different electronegativity between the atoms which are sharing the electron, that electron will spend more time over here, and this would be polar, because this is going to be a little more positive on this end, and this is going to be a little more negative on this end. So this molecule is actually polar, even though it's covalent. Now in this molecule here, so here's the polar thing. This is water. Okay, 2 hydrogens and an oxygen. So the electrons are going to sit over here more often than they are over here, which means that this side's going to be a little more positive, and this side's going to be a little more negative. So most molecules that are hydrophilic have some kind of polarity associated with them at a number of different levels. Okay, so if they have some ability to generate a polar molecule, then they will typically dissolve in water. Now this fatty acid, on the other hand, and there's carbon, 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 carbon going through here, all these carbons have relatively the same amount of affinity for the electrons. And so the electrons can actually float around here without getting the other carbons too bent out of shape. Okay? They can actually, that's why things like graphite, which is all carbon, is so greasy. Okay? It's because those electrons are free to slide. Okay? And so nonpolar molecules, you tend to have electron sharing that's very, very even across it, and that makes them nonpolar. They don't form polar compounds, and so they'll dissolve in things like oils and fats and so on. Okay? And with cell membranes, a key component of the structure of cell membrane has to do with the alignment of these polar head groups. Remember, these are phospholipids here, so you have a polar head group, and so they're going to want to associate with aqueous things. And then you have association of the nonpolar tails. Okay, so these hydrocarbon tails tend to be very lipophilic. And so the oil and water wants to separate. The lipophilic groups associate with each other. The hydrophilic groups will associate with each other, and that is the basis for what we call the lipid bilayer. Okay? Even proteins, like this protein which is embedded here, may have more hydrophobic groups residing in the outer shell of this protein, whereas there may be more hydrophilic groups here on either side of it, or perhaps down the middle, in order to facilitate its placement inside the cell membrane among these hydrophobic tails of the cell membrane. So this polar, nonpolar plays critical importance in basic structure of cell membranes. Another basic term here, you've probably heard this in your chemistry class, or you may have heard it in 165, and they call these things functional groups. And these are combinations of atoms, combinations of different atoms from different elements that we see quite often in biologic molecules. And they work together as a single unit. We sort of treat them as a single unit, rather than, say, like for instance, a phosphate group, PO4. We don't treat each oxygen individually. We treat the phosphate with the oxygens all around it as a single unit, and it has unique chemical properties that cells and molecules use for identity and for function. Okay? They provide reactivity, they can provide structure, but the reactivity is really, really important for things like interaction. How reactive is it? If it's very reactive, it's going to interact with a whole lot of things. Okay? It's going to have a much different way of interacting in a cell than something that doesn't have a whole lot of reactive functional groups. And here's a huge chart, and by no means I'm going to test you on these. Okay? This is just showing you fairly common chemical reactive groups. Okay, if you had organic chemistry, you've seen some of these. Okay, we tend to spend a lot of time with things like phosphates, alcohols, okay, aldehydes. You know, these things they confer reactivity to biologic molecules, particularly proteins. Okay, the amino acids of the proteins in the body differ by virtue of what types of side reactive groups they have. So that's basically covalent bonds. Covalent bonds serve as energy reserves. They help define polarity in polar and nonpolar. You can store energy inside these bonds, and they also are the basis for reactive groups. Okay? So one role is covalent bonds, and those roles of covalent bonds to create reactive groups and so forth. That's a key role of electrons. So now let's talk about ions. When we lose an ion, it becomes more positive. We call that a cation. I always remember the T looking like a little plus sign. If we gain an electron, now we're negative. That's a negative ion or anion. So what do ions do in cells? Well, we first defined it. It's an atom with an unequal number of electrons and protons. If it's got more protons, it's positive. If it's got more electrons, it's negative. For our purposes, ions are important because we're dealing with aqueous solutions. It's very, very unusual. It's rare that we deal with too much in terms of the lipid side of it. Yes, we're gaining more appreciation for lipid chemistry, but really most of the action is occurring in aqueous situations. It's really hard if we have a non-aqueous solvent, like say an oil or a fatty membrane, that you would see an ion inside that fatty membrane. Okay? But something about ions that we should understand. This is sort of getting to a larger purpose here. It's this statement right here, that ions are dynamic. Okay? Ions will want to achieve what we call an ionic equilibrium. So if, let's say, for instance, this phrase here is actually a cell membrane. If we have a whole bunch of ions on this side, and if there is some way for those ions to move through that membrane, they're going to want to move away from this environment into this one. That's the basis of diffusion. So one of the things about ions is that they want to achieve equilibrium in their solvent. Okay? So they're charged atoms, cations and anions. I won't spend too much time there. It's just review. We've already talked. Okay? One aspect of ions is the formation of ionic bonds. Okay? When these atoms gain or lose electrons, they gain or lose charge. Okay? That means that opposite charges will attract. So if you have a positive cation over here and a negative anion over here, they're going to want to move together. Here's some common ones. We don't see this a whole lot in biology, but it's important to understand that we want to achieve a stable outer electron layer. And so something like sodium, atomic sodium, with only one electron in its outer valence shell, really wants to get rid of that in order to stabilize. Whereas chloride, or chlorine, wants to gain an electron in order to be in its most stable energy state. So sodium's more than happy to oblige in that situation. Okay? So what happens is we lose this electron to the chloride. It never goes back to the sodium in this situation. And so this is going to be positive, it's going to be negative, and they'll be attracted by that, what we call electrostatic charge. Okay? But we see this in protein structures as well. Okay? So certain amino acids, when they're in aqueous environment inside the cells, want to become somewhat positive, just like sodium. If you were to dissolve sodium into water, you'd see that it would mostly exist as a cation. Okay? And there are amino acids with functional groups that, when they're in the aqueous situation, will form negative charges. And so now we have the basis for attraction between amino acids, which is the basis for structure. So that's one key aspect of ionic bonds in biology. It's a very, very important aspect of creating proper protein folding. If proteins are not folded properly, they can't function. Just imagine this. Imagine if you have a bicycle wheel, and all of a sudden you drive into a huge, on your bike, and you hit a huge chuck hole, and now the wheel has bent. Can it function? No, it's been improperly folded. Okay? So when you're looking at structures of proteins, one thing you want to key on to see an ionic bond is look for a little plus and a little minus. So this guy right here, not this guy, see the dots here? And not this guy, see the line between those two S's? That's a covalent bond. This is a hydrogen bond. This right here, the little plus and the little minus, may be hard to see in this figure, but you can see it here. A little plus here, a little minus there. That screams ionic bond, and it's important for proper protein folding, which means that it is important for protein function to have the right ionic environment. Now, aside from proteins, sodiums and potassium, calcium, all of these ions we're going to see at differing degrees in physiology. I guarantee you we're going to see these guys big time. These are probably the two biggest ones. pH, that's what the hydrogen ion concentration means. Okay, we might talk a little bit about chloride. We'll get to bicarbonate late in the semester. Hardly ever talk about magnesium in these guys. I will allude to some of them some places, but they not only for protein structure, we talk about folding, but these guys are really, really important for protein function. And I'll explain this a little. So we've done ions now. Now let's move to high energy electrons. Okay, high energy electrons. This is not quite as well understood, but like what I said earlier, when you impart electricity into the tube that's in the ceiling, those electrons get bumped up in an energy state, which means they move away from the nucleus of the electron. And so they've gained energy and they're moving a little bit faster. And when they lose that energy, they give off that energy, often in the form of light or heat. Here's a great YouTube video explaining this to help you understand that. That chemical bonds like covalent bonds can store energy, but we can also store energy within atoms themselves depending on what shell the electron is residing. Really, really important. This is the best example I have, but you do see it in a number of places. The energy stored in the phosphate diester bond here between, this is 1, this is 2, this is 3, this is alpha, okay, this is beta, and this is gamma. Between the beta and gamma phosphates, there's a huge amount of energy stored right here, and that has to do in part with high energy electrons. Okay, the reason adenine and ribose are used in ATP is they can help store some of that energy that we talk about in ATP. The energy is released when we cleave this bond right here, but the properties of this molecule are very, very important for being able to create those high energy bonds in the first place. Okay? And we talked about this in 165. We use high energy electrons as energy shuttles to create chemical bonds in things like photosynthesis. We use high energy electrons as energy sources to facilitate protein function. Okay, so that's high energy electrons, not as complicated as, say, covalent bonds or ions, but still you need to understand what's going on when we mean by high energy electrons. And then lastly, and this is probably the most obscure for most of us, this is called free radicals. Okay, free radicals are reactive functional groups in cells. They are highly reactive, and let me explain what I mean. Okay. So free radicals are either molecules, so groups of atoms, or individual atoms that have an unpaired electron. Okay? Free radicals are either molecules or atoms that have an unpaired electron, and an unpaired electron is a lonely soul. Okay? An unpaired electron wants to find a dance partner. Okay? It will find the nearest dance partner it can find, which means that free radicals are highly reactive. They're unstable. Unpaired electrons are not favorable energy-wise. And so if you have lots and lots of free radicals, they may be attacking reactive functional groups in proteins or in sugars or in DNA, causing damage. Now, some free radicals are very, very important for cell biology, but if we start generating tons and tons of these, that can lead to injury and disease. Okay? So our sodium atom here, this is a single sodium atom just all by itself, has that one electron sitting right here. That is a very, very reactive, a very, very unstable situation. Okay? It will react quite violently and in some ways to create molecules. Now this molecule right here is called nitric oxide. Okay? It is also a free radical. Why is that? Because it has an unpaired electron. It's a little more stable than this sodium ion over here, but it has that unpaired electron. And even though it's not charged, this is a neutral molecule. It's not charged. It has the right number of protons, or not right, but it has the same number of protons as electrons. So it's neutral, but it has that unstable, free, unpaired electron. So it's reactive. Okay? So both of those are free radicals. All right? Very, very important in things like immunity. When a phagocyte, like a neutrophil or a white blood cell, is swallowing things like invading bacteria, okay? If you have a pimple or if you have an infection somewhere, what's going on? Or white blood cells are attacking the bacteria. One of the things they do is they start generating free radicals like crazy, because those free radicals will then bind and muck up the proteins of the bacteria. Okay? If you've got just tons and tons of these, they're going to bind anything they can find. You lost control of these situations, and now they're going to get stuck in the machinery of the cell, and the machinery is proteins. Okay? So if you have a protein that has all these things stuck to it, it can't function anymore. Imagine what would happen if you had a bicycle and all of a sudden you started spraying that bicycle with something reactive like tar. Okay? Or something very sticky, like, it's hard to imagine, but imagine you just have this tar and you cover the bike in tar. Now everything's reacted with it, and the bicycle can't function anymore. That's what free radicals do. A little bit is good. In fact, a little bit is important for things like immunology. It's important for signaling, but if you have a lot of them, things go wrong. Okay? So they're important for signaling. Free radicals are important for cell signaling. Nitric oxide's very, very important for things like blood vessel dilation. If you know somebody who has heart pain, we call it angina, they take nitroglycerin. And they take the nitroglycerin to release nitric oxide, which dilates the coronary arteries. Okay? We also have drugs like Viagra. Viagra is involved in nitric oxide signaling, okay, for male sexual function. The same drug that we use in Viagra, we use in Rogaine. Why? Because nitric oxide it modulates vasoconstriction and vasodilation. So if we increase the nitric oxide on the top of somebody's head, that causes vasodilation, which can cause additional hair growth. Okay? So that's the role of free radicals. They're involved in signaling, they're involved in immunity, and they're used because they are reactive by their very nature. So we've talked about covalent bonds and ionic bonds. Remember that these are important for energy storage as well as protein structure, which then leads to protein function. Now let's move into the realm of hydrogen bonds. Hydrogen bonds are similar to both covalent and ionic bonds. They aren't covalent bonds because you're not sharing the electrons per se, but they're not necessarily ionic bonds either. They involve partial charges, so a partial negative charge here and a partial positive charge here. That's why the line between these two molecules is dotted. Okay? And hydrogen bonds are involved in hydrogen bonding with either usually an oxygen or perhaps a nitrogen. And it's very important in like DNA structure, but it's also very, very important in protein structure. Sort of to give you an example of what we're talking about with protein structure, you know that when you jump into a swimming pool, now they're here at the end of summer, or if you're taking a shower or something along those lines, your hair changes characteristics. Your hair generally gets less curly, it gets more floppy, more smooth. That's because the curliness and the general structure of dry hair uses hydrogen bonds. Okay? Now, if you have extremely curly hair, like for instance an African American or somebody who has naturally very curly hair, there are some other bonds there too. But for most of us who have wavy hair or relatively straight hair, hydrogen bonds are at play in dry hair. And as soon as water comes in, these hydrogen bonds get released to a certain degree. Okay, and that's what makes the hair more floppy, more smooth. That's what makes something wet, is oftentimes you're getting the mitigation of hydrogen bonds. So you can recognize hydrogen bonds by these little dotted lines, and you don't see a minus here and you don't see a plus here. So that's how you can tell hydrogen bonds. The last category of bonds here are called van der Waals forces. We also have some other bonds called hydrophobic interactions. We will lump those all together into van der Waals. I might call them van der Waals / hydrophobic. But let's get back to our graphite here for a second. So here's a layer of graphite, here's a layer of graphite. The reason these layers slide across each other is that these bonds tend to be very, very weak. Okay, so that allows for slipperiness. It's because they're nonpolar interactions that are a very, very weak sharing of electrons. The electrons are free to move around. That's why graphite can actually conduct electricity. So van der Waals hydrophobic interactions are the weakest, but they do play a role, and we'll talk about them a little. So again, hydrogen bonds are an electrostatic association. It's hydrophilic, but they're neither covalent nor fully ionic. That is to say, they do have charge. It's just not a full charge like in an ionic situation. Usually you can recognize them because it's either oxygen or it's nitrogen bonding with a hydrogen atom. Okay, like in this situation, you have an oxygen right here. It has a little bit of a negative charge. This is a polar molecule. It's a little bit negative. The electrons are more out here, and they are associated with a partial positive charge from this proton right here, forming this dotted, dashed interaction between the oxygen of one molecule of water and the hydrogen of another. So the question I will pose to you is, does this oxygen care what hydrogen it bonds to? And the simple answer is no. If there's another hydrogen out there somewhere that may even have a little more positive charge, it may stand in for this hydrogen right here. Okay? Setting you up for something a little bit down the line. But again, here are some nitrogenous bases, and here's a hydrogen bond, here's a hydrogen bond, here's a hydrogen bond. So it's very, very important in DNA structure, and to a certain extent RNA structure. But it's also important for protein folding. And where you see lots and lots and lots of hydrogen bonds are in what we call the secondary structure. I'll get to that in a little bit. Primary structure is the chain of amino acids, and then when you begin folding it in what's called secondary structure, either a helical situation or a sheet, if you will, that's primarily mediated by hydrogen bonds, and that's what makes them extremely important. Okay. So with van der Waals forces are very weak. Okay, they are non-polar, and they usually occur in sort of hydrophobic situations where electrons can sort of flow around and create these very, very minute changes in charge density. Okay, so we often have what are called hydrophobic interactions inside proteins that help stabilize structure, but they're usually very weak. Okay, they are not covalent and they are not ionic, but because these electrons can sort of flow around. All of these atoms here are carbon atoms, so these electrons form sort of a cloud, if you will, and those clouds are very, very loose. So this layer here, which is connected with covalent bonds, very, very strong, is connected with van der Waals forces between the next layer, which means that these two layers can slide against each other quite easily. So you might pull on the fiber of graphite and it won't move. It's very, very strong, like a carbon fiber, but at the same time, across the layers, they may be very, very slippery. And that's why graphite is often used as a lubricant. It's important in things like cell membrane structure. These hydrophobic fatty acid tails are interacting with each other through hydrophobic interactions and van der Waals forces. This is a picture here from a research article where they took a protein that is in this shape, and they could predict what would happen to it if they started increasing the pressure. I'm not sure if it was gas, or they were changing the conditions, so it's getting more and more severe as you go. And what they found was that when they predicted the protein structure, accounting for these hydrophobic interactions, as you went down here, if the hydrophobic interactions were in place, you actually had more of a structure that was similar to the original state than if you didn't account for hydrophobic interactions. You can see that this guy over here is much more screwed up than this guy over here. So maybe there's a little bit of function, even though this is a severe condition. Or if you look at the 10 condition, you can see that this one's much more intact than even this one over here. Okay? So it does play a role. It's just a very, very subtle one. And that's something that we don't have our brains wrapped around entirely yet as to the overall importance of those hydrophobic interactions. We know they're important when protein subunits are linking together, but it's still a very subtle thing, and it's hard to just give you a this or that kind of an explanation. And so these chemical bonds, the four major chemical bonds, so we have covalent, we have hydrogen, we have ionic, and we have the hydrophobic / van der Waals forces, contribute to the three-dimensional structure of proteins that we observe in nature. So this is a kind of a tube of helices. So if you're looking down the barrel of the tube, you can see that these helices are forming this barrel. And we were studying this modification right here in red, and you can see that it's sticking right down in the middle of this thing, causing impacts on these other helices. Okay, it was a subtle modification that was changing the function of this protein. So let's talk about how these bonds specifically affect protein structure. So we have the covalent bonds, most common when we talk about proteins, when we're talking about folding. Now, if we look at the primary structure, if we were to follow this ribbon here, what links the amino acids together in this ribbon is a covalent bond. We call that a peptide bond. Well, what's going on here is that we've got this loop here, and these two amino acids, these are different amino acids here, their side chains have been linked with a covalent bond, and this is a very common one called a disulfide bond. And this is the bond that we break when somebody has their hair straightened. So if you've ever seen a movie where they apply very, very strong chemicals in order to straighten hair, what they're doing is breaking those disulfide bonds. They're very strong chemically, so they require strong chemicals to break them. Or if somebody has their hair permed, what we're doing there is we are artificially creating disulfide bonds or covalent bonds between different amino acids in order to get that sort of curly structure in the hair. Okay? Another bond that's very common is ionic bonds, and you can always spot them because you'll see a plus and a minus in there. This is a negative, this is an anion, and this is a cationic amino acids, again stabilizing this particular loop of this protein. Okay? Then we have hydrogen bonds. They can all do the same thing to varying degrees of strength. For instance, this bond right here, because like I told you earlier, this oxygen doesn't care if it's binding to this hydrogen or some other hydrogen. So this loop right here may be more vulnerable to changes in pH or changes in how much water is present. Okay? This one right here may be more vulnerable to changes in ionic concentration in the solution. Because why? Because this guy doesn't care what positive charge he's linked to. So if we had another sodium, a positively charged ion come in, it might release this association to allow this to straighten out. Okay? And covalents are usually very, very strong. So we find a lot of covalent bonds in very strong proteins. We also see them in environments that are relatively extreme, like hot environments that are very, very high temperatures or very, very low pHs. Okay? And so that's what we're showing down here. We've got an ionic bond right here. Here's another ionic bond. Here's a hydrogen bond. And here's a hydrophobic interaction. There's our fourth association. Here's a hydrogen bond here. So you can begin to imagine some of the possibilities of folding and structure through these four bonds. And I'd like you to be able to identify the level of structure. There's primary, secondary, tertiary, and quaternary, and what is the most likely bond to be involved in that particular level of structure. Okay? So here's primary structure up here at the top. So these are amino acids. This is the linear sequence of amino acids translated from an mRNA sequence. And so they are linked by what are called peptide bonds. Those are covalent bonds linking one amino acid to the next. Okay? The next level is what we call secondary structure. In this case, it's a helix, like we've shown here. See this kind of helical shape here? Or sometimes it can be a zigzag sheet, if you will. We call this alpha helix, and we have the other major structure is called beta sheet. Okay? And those are normally stabilized. Secondary structures are normally stabilized by hydrogen bonds. Okay? Tertiary structure. Now, if we take this helix here and fold it in another direction and fold it in another direction, fold it this way. This is almost all helix here. We're not seeing a whole lot of beta sheet in here. That's called tertiary structure. So what would normally be a straight helical structure gets bent around and reorganized and folded back on itself. That's known as tertiary structure. And we see covalent bonds, we see hydrogen bonds, we see ionic bonds, and we see hydrophobic van der Waals forces. We see all four types of bonds in tertiary structure. We normally just see hydrogen bonds in secondary structure, and we normally just see covalent bonds in primary structure. The last structure down here, this is the fourth level. This is an independent peptide down here, independent peptide here. These are not linked with the normal primary linkage this way. These subunits are associating together, and what we see here is anything but covalent bonds. So we would see hydrogen bonds maybe between these two here, or we would see ionic bonds between the subunits and among the subunits. Okay, and we also see hydrophobic van der Waals, but we normally never see covalent bonds between these subunits. Okay, so one more time: covalent only, hydrogen bonds primarily in the secondary structure, alpha helix, beta sheet. Bending of secondary structure and folding of secondary structure creates tertiary. That's all four bonds. Okay, covalent, ionic, hydrogen, and hydrophobic van der Waals. And then lastly, in quaternary structure, we see ionic, hydrogen, and hydrophobic van der Waals. We do not see covalent. So molecular shape and function. Again, shape drives function. Think about, for instance, a pair of scissors. Okay, the shape is very, very important to that pair of scissors function. Same thing with a bicycle. If you have a bicycle with a bent wheel, it doesn't have the right shape, it doesn't have the right function. Okay, so our molecular bonds, our covalents, our ionics, our hydrogens and hydrophobics determine shape, and then shape drives function. Okay? Proteins are extremely complicated that way. They have very, very subtle shapes. They wiggle a little bit. I think I have some videos of that coming up. It's all about the wiggle. It's all about the direction of the wiggle. Again, let's think about a pair of scissors for a second. If the scissors are not wiggling, if the blades are not wiggling past each other, if they're wiggling, let's say, in perpendicular fashion to what we talked about, they're not really going to cut anything at all. Okay? So shape is everything. Shape drives function. Okay? So the shape of the protein drives its function. So if an enzyme has the right shape, it will catalyze a reaction. And if that shape starts to go away for any reason, because of temperature, because of pH, because of ionic concentration, then the ability to catalyze the reaction goes away. And the same is true for all of these. Okay? All of these here are examples of soluble proteins. Okay? These proteins are soluble. We don't really see any structural proteins here. Structural proteins are typically insoluble. Okay? But that proper shape depends on the chemical bonds. So if we start disrupting ionic bonds with too many ions or too few ions, you don't have enough ions, maybe the bond is too tight, and now the protein's all bound up on itself. Okay? If you have too many protons, now the pH is too low, and now the protein starts changing shape because now those hydrogen bonds are being disrupted. Secondary, tertiary, and quaternary structure. If you don't have enough, if the pH is too high, now amino acids are associating that shouldn't be associating. So there's a window, there's what we call a homeostatic window that drives whether the protein is properly shaped in order to achieve its function. Okay? And one of the most common and among the most important is pH. pH is hydrogen ion concentration. It's the logarithm of hydrogen ion concentration. Okay? Because secondary structures are important and they're driven by hydrogen bonds, tertiary structures are important for shape and driven by hydrogen bonds, and quaternary structures are important for function and hydrogen bonding. So that's why how many protons that are freely available in that solution can so dramatically affect a molecule's shape. Okay? So pH is the inverse logarithm of how many protons are in the solution. Okay? What we find, and I'm going to try to, it's not well illustrated here, but what we find, if you look at a neutral pH, and you guys should know what neutral pH is, that's 7.0. What does that mean? 7.0 pH means that one molecule in 10 million water molecules has lost its proton, and that proton is free. Okay? It is 10 to the negative 7th. That means 1 in 10 million. So if you have neutral pH, that 1 in 10 million molecules has ionized to form H plus and OH minus. Alright? Now 1 in 10 million sounds like pretty long odds, doesn't it? But when you're talking about 10 to the 23rd molecules, 10 with 23 zeros behind it, a 10 with 7 zeros behind it is not that. It's pretty often. Okay? That's fairly common. Okay? In our human body systems, most of the time, with maybe some exceptions in the cerebrospinal fluid, and definitely some exceptions in the digestive tract. If you think about stomach pH or certain pHs in the small intestine, the magic number for our bodies, for our cells, is 7.4. That means we're a little bit on the alkaline side. So kind of keep that number in mind. Anything lower than 7.4 means that we have more free hydrogens. Anything higher than 7.4 means that we have fewer free hydrogens, and it's a factor of 10 for every whole digit. So 8.4 pH means that there is only 1/10 the number of free hydrogen ions. 6.4 means that there are 10 times as many free hydrogen ions. Okay? It doesn't take much to screw things up. Why? Because of that secondary and that tertiary and that quaternary structure. Those hydrogen bonds will be disrupted if you have either too many free hydrogens. That's easy to visualize, but also if you have too few. Okay? So if you have too many free hydrogens, you're acidotic. Okay? We call that acidosis. Respiratory acidosis, metabolic acidosis. Okay? Your body's pretty good at handling this. On the other hand, if you have too few, we call that alkalosis, and that is a tough one for the body to handle. It's much more able to handle acidosis because most of our organic molecules have an acidic tendency to them. The alkalotic means that we have too few free hydrogen. The proteins are no longer shaped properly and they're beginning to become unregulated or stop functioning altogether. Okay? So we have lots of buffers. We'll talk about one or two okay, in class, primarily carbon dioxide and what we call bicarbonate. But we'll get to that in due time. But I want you to keep this number in mind, understand the relationship, understand what it means to jump a whole number. So if we go from 7.0 to 8.0, 7.0 to 6.0, and understand why. I really want you to get at the why. Free hydrogen, either too much or too few, can screw up proteins, and why in acute care settings we spend so much time focusing on this. And if you've forgotten your acids and bases, here's a little review. I just have I'll leave this slide here. I won't talk about it too much. I've already spent a lot of time already. Review this. It's in your book. If you feel that you're a little weak on this, come see me. We can work on this. But this is important for things like the MCAT, if you're going to take the MCAT, or the GRE, if you're going to take that. It's a review on acids and bases, and if you've lost track of it, let's work on it, either during discussion or free time in the lab. So a little bit of a review. Take a deep breath. I really want to encourage you to try to imagine and think about some of these things on your own when you have a free moment or two. I know that we live busy lives and you're talking to people and texting and you got lunch and that sort of thing, but take some time and think about what we've just talked about. We've talked about molecules and bonds, and the role of electrons. We had 4 major roles of electrons and functional groups, the relationship of shape and function. We talked about non-covalent interactions, which include ionic bonds, hydrogen bonds, and the hydrophobic van der Waals. Okay, where do we find these, and what are they doing? And why would they be screwed up with things like concentration of ions like sodium or chloride or calcium or potassium? Why hydrogen bonds would be screwed up because of pH? And then we're going to now talk about protein interactions. How proteins behave, why they do what they do, and how we can predict what they'll do if we start changing the game on them. Okay. So we talked about this a few slides earlier. Soluble proteins, these are the real functional proteins. They don't do a lot of structure, but they do a lot of busy work in the cells. Okay, they are catalytic enzymes. Okay, things like protecting your cells from oxidative stress, or converting glucose to ATP. We have membrane transporters. These guys regulate what's getting and going where. For instance, very few things get through the cell membrane. We tend to assume that water's going to fly through there, but the truth is you have to have a transporter to let that water through, or the water's not getting there. We have to convey information through the cell, from the outside of the cell to the nucleus. Okay, we call that signal transduction. We have to perceive changes in the environment or signals coming in. We call those proteins receptors. We have chaperones and carriers. We call these binding proteins. For instance, the steroid hormones that are flowing through your body right now in your bloodstream are typically stored inside of a protein as they float through the bloodstream. Immunoglobulins, these are the antibodies. Okay, we have 4 or 5 different types. They're involved in the immune response. Okay, immunoglobulin, so a soluble protein that's involved in the immune response. And then lastly, we have these things that will bind DNA. Okay, or they'll bind other proteins and control them. They'll act as brakes, or they'll act as accelerators. We call these regulatory proteins. Their job is to either increase or decrease and control the rate of other things like, for instance, catalysts or transporters and so forth. They are controllers of the effector molecules inside cells. So we have soluble proteins. And then we have insoluble proteins, and most of these guys are structural proteins. Collagen, fibronectin, laminin, keratin. If you want to feel a structural protein, just grab a little bit of hair. That's primarily keratin. Feel your fingernails, same thing. Primarily a form of keratin, very similar to rhinoceros horn. But these proteins interact, particularly the soluble ones. The soluble proteins are very, very interactive. Okay, and that interaction we typically call binding. Okay, binding is a non-covalent interaction Okay, so it's going to involve hydrogen bonds and ionic bonds and hydrophobic interactions with other molecules. They can be non-protein molecules or they can be other proteins. And they typically occur at very specific sites, and we call those places binding sites. And the reason we're so concerned about that is that we may find molecules that we can either design or we find them in nature that interfere with that binding site. Kind of like sticking glue inside a lock. The lock, at that slot that you stick the key in, would be considered a binding site. Okay? So we have a binding event, okay, at a binding site, and those binding events are very similar to non-organic chemical reactions. They will want to move to a state of equilibrium. Let me say that one more time. They will want to move to a state of equilibrium. Very seldom will we let it get to a state of equilibrium. They'll try to get there, and we may let it move that way, but we're going to deal with it so that it never does. There are very, very few things in our body that are actually at equilibrium, and if we reach equilibrium, we're either in disease or we're dead. Okay? So when we have binding events, if a protein is binding to something, we call that molecule or other protein a ligand, okay, or a ligand if you're British, okay. If it's a catalyst, like a protein that is catalyzing a chemical reaction, sometimes we will refer to it as a substrate. So what's going to happen to this substrate? It's going to be affected by the protein. But ligand or ligand and substrates are a somewhat interchangeable term. We tend to use substrates as in catalytic situations. We tend to use ligands in non-catalytic situations, in regulatory proteins or receptors and so forth. Okay? And proteins are specific. They like certain shapes of molecules. But they're not exclusive. So if you have another molecule, and pharmaceuticals are very, very much like this, the pharmaceutical, the drug, may bind a protein because it looks like a ligand or a substrate. Okay? So proteins are specific about what they will bind to, but they're not exclusive. And that's why pharmaceuticals can work, because they can mimic the natural ligand or the natural substrate. And so here, now's the time. Remember this link I showed you earlier? This is a really, really good link. It would be worth your time. If you're feeling uncomfortable about this, if I'm not making sense, if I'm moving through this too fast, you can go through this. This is an online course, and they say it takes about an hour. So if you want that edge, you want to get a little more, from a different perspective than Dr. Wiseman's perspective, by all means use this resource. It's a good resource, and I will be thrilled if you find that you can learn from sources other than my voice and my lectures. I think that's good. You're learning independence and you're learning self-sufficiency. That's great. So here's an example of a ligand that's binding in the binding site of a protein. Okay? That ligand wants to be in that spot because it seems to have functional groups or a shape that facilitates non-covalent interactions. Almost always when a protein is binding to its ligand or its substrate, it is specific, it is reversible, it will obey kinetic laws. Okay? So if you have a whole ton of this, it will tend to push it in this direction. If you have hardly any of this, the reaction will move back in this direction. Okay? But importantly, it's non-covalent. It's very, very rare that you have a covalent interaction. I'll give you a classic example of an exception to this. Aspirin. Aspirin covalently modifies the proteins that are responsible for creating pain signals. Okay? It's one of the rare exceptions. Advil, ibuprofen, on the other hand, is a non-covalent inhibitor of those same proteins. Okay? So it's specific, it's reversible, it wants to move to equilibrium. Okay? We're usually not going to let it, but it will happen. It'll try to get there. Okay? And it's non-covalent. Okay? We've already talked about this. This is just rehashing. Okay, ligands and substrates are specific. Okay, we call this specific non-covalent interaction. We call that molecular complementarity. Okay? And there are examples of some associated terms. We often refer to that complementarity as affinity. If there is a highly complementary structure, it may have what we call high affinity, which means that it's going to move to a more often to a bound state than an unbound state. This induced fit model, I'll show you what I'm talking about. So here's an induced fit model. What it's saying is that by binding B1 here, let's say this is the binding site for B1, or B prime, I think it's B1. This binding site comes in here, this is substrate 1, and it changes the shape of the protein. You can see how the protein has now flipped over like a garage door closing on B1 here, and that change in shape now allows B2 to come in. B2 may never bind to the protein, or very, very rarely bind to the protein without first B1 binding. And so that's what we call induced fit. A way to sort of explain this would be almost like a baseball glove closing around a baseball as the ball flies into the... Here's another example of an induced fit model. Here's ligand 1 up here. Okay, here is ligand 2. They each have individual binding sites. Unfortunately, L2 cannot bind until L1 binds first. Okay, this has nothing to do with affinity. Maybe right now, well, I take that back. Right now, there is very low affinity for L2. When L1 comes in, it changes the shape, however subtly or however emphatically, it changes that protein shape, and now the complementarity has increased and the affinity has increased, so it's much more likely to allow binding to occur. And so protein binding and unbinding and associating with ligands and ligand-like molecules obeys something that we call the law of mass action. Okay? The idea with the law of mass action is that normally in aqueous situations, and we might as well just assume it's aqueous, reactants that are unbound will want to move between a bound and unbound state. That's what's known as equilibrium, when unbinding occurs at the same rate as binding. Okay? Very, very unusual to see that in biology, but it's still an important concept. So that we have P plus L here. If you have concentrations of these, they will begin to associate into a conjugated bound form. Okay? This could be two chemicals doing a chemical reaction. It could also be a protein and its ligand. Both of those scenarios obey what's called the law of mass action. The idea is that you're having an interface between bound and unbound, but it's not equal. And we can actually measure that. It can actually be very, very skewed to one side or another. For instance, if we had sodium over here and chloride, and we put them together, they will violently react and almost completely form this. So the ratio of this to this in that situation would be very, very high. Most of the time in biology, it's a little more even-handed, that you're going to have a certain percentage which is bound, and a certain percentage which is unbound. And that's what this equilibrium coefficient is right here. This is a number that we use, it's really shorthand is all it is. It's really shorthand of a ratio. If you have a really, really high number here, that means that most of it's all bound. That's all it means. Okay? So we go through some of this mind-numbing math sometimes in order to explain relatively simple concepts. The concept is that this reaction wants to reach equilibrium of some kind, but it's not necessarily even on one side or the other. It's a ratio that's arbitrary for the protein, for the ligand, or for the reactants in the product. Okay? Let me explain something else. If we start changing the amount of reactant or the amount of product, that causes a change in the mass balance. And so the law of mass action means that we want to get back to a more balanced state. So if we start adding mass here in the form of unconjugated protein or ligand, that will increase the drive to push it to a bound form, because we always want to reach that KEQ. In this state right now, the unbound relative to bound is less than that calculated KEQ, and so the reaction is going to want to proceed in this direction. Okay? What we normally do in physiology is that we never let equilibrium be achieved. Okay, let's say we're trying to create something here. We're trying to create some conjugated product. So we react stuff and it starts moving in this direction, and then we kick this out. We either turn it into something else, or we move it away from the site of reaction, some other place in the cell, some other compartment, or we kick it out of the cell altogether so that the reaction can only proceed in a given direction. Or vice versa. Let's say there's another reaction out there somewhere. We're trying to break stuff up, a catabolic reaction, if you will. So we'll bust it up, and now we'll have these reactants, and we'll ship those somewhere else, convert them into something else so they can't react. That's the law of mass action, and explaining to you that scenario that reactions want to achieve a ratio is something that cells and proteins exploit in order to get things done inside the cell. Okay? So here we go. So we are going to push it this way because we're less than KEQ, so it's going to want to go this way. Okay? It can be proteins and ligands, or it could be some other molecules that will react with each other to form some product. Okay? Once we've restored equilibrium, then the reaction is moving at the same rate in both directions. But this is very, very rare in physiology. It's very, very rare. We live in a state of dynamic disequilibrium. Okay? Dynamic steady state, we call it. Okay? So yet, the reaction wants to get here, but we're not going to let it. Okay? Now that's different than what we're talking about here, which is affinity. Okay? The KEQ is not the same as KD, but they're calculated in similar fashion. Okay? What we're talking about here is how strong the affinity is for a protein or a reactant for its substrate. Okay? And it's a ratio just like before. It's just a different kind of ratio where the unbound concentration versus the bound concentration. So if you have very little unbound concentration versus bound concentration, that means the number is smaller. So for your purposes, when we're talking about KD, dissociation constant, okay, the opposite of affinity is dissociation. Okay? The smaller the dissociation constant, the stronger the affinity. Okay? And we can modulate affinity. We can modulate affinity with inhibitors or competitors. We can also modulate affinity with activators. Okay? But fundamentally, KD is a ratio of unbound ligand to bound ligand. It's almost an inverted reaction to the KEQ. KEQ and KD are almost inverse of each other. Okay? KD is much more common in protein chemistry. We're not going to talk a lot about KD, but I want to give those of you who will be moving into biochemistry, or those of you who will be moving into pharmacology, some awareness so that you have a place to begin when the time comes. But understand that with KD, the smaller the number, the stronger the affinity. And I think that's it.